biologists were pretty darn good with their microscopes at figuring out how many cell types there were. But what we have now is the full molecular portrait of those cell types. We have a complete molecular definition of what molecules they're using and how they work in a rich tapestry of detail that wasn't available before.
This summer, Stephen Quake took over from Corey Bargman as the head of science at the Chan Zuckerberg Initiative. Quake is Lee Otterson Professor of Bioengineering and Professor of Applied Physics at Stanford. He has pioneered multiple technologies for everything from DNA sequencing to non-invasive prenatal screening to microfluidic automation. We talked about open science and preprints, what we've learned after a decade of molecular cell atlases, current challenges in imaging, a very personal genomic project, and more. Welcome to the EMBO Podcast. Steve Quake got his bachelor's degree in physics and a master's in math at Stanford, and went on to do a PhD in theoretical physics at Oxford.
I became interested in biology as an undergraduate, actually. I did a senior thesis with Steve Chu, who at that point in time was doing both physics and biophysics. In his lab, he was working on cooling and trapping of atoms, which is what led to his Nobel Prize, but he was also working on optical tweezers and using radiation forces to trap molecules and stretch them out and measure forces. And it's what got me into biology and biological problems, and I've been fascinated ever since.
For your own lab, do you recruit a lot of students from physics and mathematics backgrounds?
Oh, yeah, I have a complete mixture. I get students from physics, applied physics, bioengineering, biochemistry, occasionally biology, chemical engineering. I've had students come in from what are the biological sciences? Biochemistry mostly, because they're quantitative enough that they kind of connect to the culture. But genetics.
And they mix harmoniously?
They do. Everybody has something to learn from someone else. Nobody comes in knowing everything they need to do to be successful in the lab, and so there's a lot of helping out and sharing expertise.
And in terms of education, is there something from that you think should be applied in the way we teach either physics or biology at the undergraduate level?
Well, yes. I mean, I think to be successful in modern biology, you need to be numerate in a way that you didn't historically. And I think you're seeing that curriculum evolving a little bit. And on the physics side, it's not just about steam engines and ideal gas law anymore. Physics, the frontiers of physics in some sense can be found in biology, understanding non-equilibrium systems. And so the examples one studies using the powerful sort of formalism and intellectual machines of physics should evolve.
So previously, I worked at the Gulbenkian Institute, and we had some experience trying to put together a graduate program for bioinformatics that mixed people from a math background and from a biology background, and it did pretty well. But at the end, one of the great difficulties was that it seemed to be a lot easier at the graduate level to teach biology to people from a quantitative background than vice versa. I think a lot of that also is because you can pick up a lot of biology ad hoc. As you become interested in the mitochondria, you don't have to understand the endoplasmatic reticulum so much. You can take your Legos and assemble them, whereas quantitative skills have to be built perhaps more like a language, in the sense that each step is the foundation for the next. And recently on the EMBO podcast, we spoke with Eugene Koonin, and in the end, I don't think we used this in the just for time reasons. It wasn't in the final edit of the podcast, but we were discussing the educational issue, and his point of view was somewhat bleak. I think in that he said that the quantitative tools had grown so much, and the bioinformatic tools had grown so much, that it would be most advisable for people from a pure biological background maybe to rely more on off-the-shelf tools and collaborators than to jump in with both feet and try to develop that expertise on their own. Would you agree with that?
I'm eternally an optimist. And, I just think that, especially as a university professor, it all comes from the training and the curriculum. And so I think the nature of biological training is changing, and people who are going to have those tools as they come along. And one of the things I did at Stanford was to help start the bioengineering department. And that was the only department at Stanford jointly owned by two schools, both engineering and medicine. And we were essentially the first undergraduate major that the medical school had. And the dean at the time, Phil Pizzo, decided this is incredibly important for the future of the medical school because they needed doctors who were numerate. And we designed the undergraduate curriculum in bioengineering so that one could go to medical school from there, precisely for that reason. And we've seen a number of very successful grads go on to do amazing things over the years. So I think this new generation we educate is going to be fully bilingual.
I like the optimism. I hope it pans out. And when you got to the end of your PhD in theoretical physics, and then when it came time to move on as a scientist, how did you pick the area of biology you would be working on?
Yeah, that's a great question. So since I didn't really move on because I went back to Steve Chu's lab to sort of finish what I'd started as an undergraduate. And, I think maybe the way to answer your question is my first year as a graduate student, I spent in the library just reading. And I decided I'm going to survey all of physics, see what's interesting to me. And I kept coming back to biophysics. And within that, I was most excited by what was then the emerging world of single molecule biophysics, measuring forces, not having to do ensemble averaging, learning about individual trajectories of molecules. It just seemed like the most exciting thing in the world to work on. And so for me at the time it was, and that's what I did.
How did your classmates in graduate school feel about that conclusion that the most exciting part of physics would be at the frontier with biology?
Oh, they all thought I was nuts.
One field where Quake and his team have pushed the envelope is in the development of molecular cell atlases, moving well beyond transcriptomics. In 2017, they published the murine molecular cell atlas, Tabula Muris, with the Tabula Sapiens coming out in 2021. In between, as Quake wrote in his 2022 Trends in Genetics opinion piece, A Decade of Molecular Cell Atlases, the floodgates for whole organism atlases opened.
From your point of view, as you've also written a lot about the artificial separation between applied science and basic science and the need for cross-fertilization between translational research and basic research. In these last 4 or 5 years, when you look at the results from these projects, was there any surprising biology for you in the basic sense, something in terms of how the organism functions or physiology that came out of these data sets that you were really surprised or just wowed by?
Well, there's a ton of stuff that's emerged, and just to sort of put it in context, I think, there's this metaphor in the lay press that the genome is the blueprint of the organism. And it's really not for any multicellular organism because pretty much every cell has the same genome, but they take on much different phenotypes. Neuron from cardiomyocyte, from, pneumocyte in the lung, whatever, you can go on and on and on. They're very, very different, yet they all have the same genome. And why is that? Well, I think the correct metaphor is that the genome is a parts list, and each cell type uses different parts. And what these atlases have done is articulate that and give us the real blueprint of which parts are used in which cell type. And there's just a ton of biology to discover there. And, for some specific examples of what's emerged that was surprising and interesting, one thing is, I think, that the number of cell types is not growing exponentially. At the beginning of this field, a bunch of people were saying, we've only discovered a small fraction of cell types. And it turns out that's not really the case. You can look at pretty much any paper anybody's published in this field, and you will not find an example where they've doubled the number of cell types of that tissue. And I'll put the brain aside for a minute. And the brain is more complex. And that one, yes, it is true, we're discovering new cell types. For every other tissue in the body, nobody's doubling the number of cell types. They're rather finding one or two new ones among dozens of cell types. And so, that's interesting because there was a lot of disagreement about that at the beginning. And, the biologists were pretty darn good with their microscopes at figuring out how many cell types there were. But what we have now is the full molecular portrait of those cell types. We have a complete molecular definition of what molecules they're using and how they work in a rich tapestry of detail that wasn't available before. So what else is interesting has come out of it? The extent to which truly transcription factor gene expression will define the cell types. I mean, better than anything else at defining the cell types. And we can say that with some degree of rigor and precision now. What's emerged? The extent to which splicing varies across tissues and cell types, and the number of novel splices that are used in humans, for example. It's much more intricate than people had realized. And even well-studied canonical splices are used quite differently across different cell types and different tissues of the body. And that is like, wow, there's a ton to bite off there. We're learning a ton, and we've just scratched the surface of this, about the evolutionary relationships between cell types across organisms. And that's going to be such an amazingly rich vein to mine for the next decade. I'm super excited about seeing where all that goes as clever people get into that. I think in specific questions we've looked at, like aging and parabiosis, we've learned just a ton about the molecular details of senescence, of change of immune repertoire with age, of the genes involved in senescence, what defines that, what can be reversed and what can't. I mean, the parabiosis experiments, I was very skeptical of that going in. I thought we were going to show that this rejuvenation thing was all artifact, and it was just people fooling themselves. And yet there really does appear to be a molecular basis for many of the effects that's true and maps onto acceleration and reversal of natural aging. Yeah, so those are a few of the things.
It was a fascinating starting point to your answer, this realization that maybe we do have a reasonable handle on how many different cell types there were. One of your statements at the outset of the project was no one really knows how many cell types there are, and it to me it gave the project an interesting spin, because usually when we see these kind of data sets, they're already partitioned by cell type. So we're going to do the memory T cell single cell atlas, and we're going to do the cardiomyocyte single cell atlas. So that doesn't really lend itself to asking the question, right? How many cell types there are? It does put one interesting problem forward also at the sort of almost philosophy of biology level, which is what is a cell type versus a cell state versus a transient alteration versus an activation state versus a developmental stage, and so on and so forth. But clearly there is a cell type. If you're saying that the morphology and the classical histology basically, obviously with many exceptions, because it's biology, maps onto what you're seeing in what would be more of an unbiased approach, it tends to indicate that what we classify as cell types are real entities. Would you agree with that?
I would. I have sort of two responses to the points you just raised. The first is, when we organized at Stanford one of the very first human cell atlas meetings, and we had a lot of people show up. That was the first one where we really opened the doors to everybody, and there were, I don't know, more than 300 people there. And I asked everybody to write down on their name badge their best estimate of the number of cell types in the human body. And you would be surprised how many people, didn't want to play the game, but the numbers were all over the map. I mean, the textbook number is like 200 or 300, and a few people were down there, and others were like, uncountable millions, and it spanned the whole thing. I mean, nobody really had any idea. There was no consensus. And I did a sort of principled calculation to get to my estimate. Being a physicist, I like to calculate things. All right, I'm going to calculate this. And I came out, at a few thousand. And the calculation was, that there's no way that biologists have missed 95% of what's out there. I mean, they're too good, and too much effort's been paid, and, we understand so much. If they've only discovered 5% of the cell types that are there and missed 95%, where does that put us? Well, the textbook number is 300. So you multiply that by 20, 6,000 cell types. That was my principled estimate. And it's probably not too far off from where the literature is going. The new ones are going to be dominated by the brain, and and most other things we're struggling to push 1,000. And so it's really going to come down to how many new cell types we're finding in the brain. That's the first point. Second point is this question you raised, which is a very deep one, about what is a cell type and how does a cell type differ from a cell state. And that's something, and I don't think the field has really come to a rigorous definition on. And, it's very operational. It's there for more than 24 hours. It's a type and not a state. And, there's a little fuzziness in that I've never been comfortable with. The other aspect of that that's super interesting is just this notion of, are all cell types discrete? And it turns out they're not. And you can see continuous variation of cell types. And we spent a bunch of time looking at that in different contexts, particularly in the striatum in the brain. But this notion that all cell types are discrete, I think, is a false one. Some of them are, but there's others that vary continuously in their properties. And we've got a glimmer of understanding about that, but I don't think it's fully integrated into the way people think about the field.
In some ways, it's almost reminiscent of the species definition problem, where there's a real biological entity there, but once you start trying to formalize it, and it becomes very, very, very tricky. But it is interesting to see this confirmation, and it really does echo the sort of thing you saw after the human genome came out. And speaking of which, of course, you at one point famously did your own genome before it was so common to do, in I believe 2009, 2010.
I was maybe the fourth or fifth human genome sequenced.
And you mentioned at the time that you were hoping to gain some practically useful information. Obviously, there are privacy and medical considerations and so forth. But is there anything you could share that was, that as you had discussed at the time, open-endedly, that would either lead to lifestyle changes or adaptations, or you mentioned peanut allergy concerns for your kids. Is there anything that of course, respecting the limits of privacy, that you could share from that?
Yeah, I mean, not a lot of privacy in this, in that, when we had that done, I was like wandering around Stanford, knocking on doctors' doors saying, help me understand this. And my colleagues got together and they got very interested in this idea of how do you treat a patient who's got their genome in hand. And they ended up, a whole team of them, led by you and Ashley and Russ Altman and Atul Butte, doing kind of a deep dive and wrote a paper in the Lancet.
Oh,
this is how you do it. First clinical annotation of a human genome. And what came out of it on my side, as the patient, as it were, was, I think, two things. One is that we all have a few skeletons in our genomic closet, right? The estimates are everyone's inherited maybe half a dozen, let's call them not so positive alleles. And the genome sometimes helps you get at that. In my case, there was a new, previously uncharacterized mutation in a gene that's been associated with cardiomyopathy. And that kind of got the attention of the cardiologist and said, hey, maybe you should be, kind of screened a little more often for cardiomyopathy than somebody who didn't have that mutation. And so those kind of rare things help you think about what you should keep an eye on as you age. The other thing that was very interesting, I think, and useful was the pharmacogenomic analysis, where they went through and gave me a list of drugs and said, these you'll respond to. These may have side effects. And, that's very practical information. And when it came time for me to go on to statins, because my lipid levels were not optimal, a little easier knowing that, hey, I'm a good responder with low side effects to some of the statins. And that made it a little more comfortable for me to make that decision to do that.
I have to look for that Lancet paper. I once ran into a guy in a wine bar in Porto who was very proud that he had actually been a case study. And I don't know how we got to talking. And at the time I was an editor, and he said, I've been in a medical journal. I am the patient in this case study. And he went to get the article. And
yeah, it was a funny thing. The genetic counselors had their heads in their hands the whole time, because this is not what they were used to, right? Because I was like the world expert in the quality of the genome, because I had done the sequencing, the assembly, and I knew how much confidence to have in various things. And as they were poring over things and trying to sort through, is this real? Is this artifact? I was the one they had to ask. And the whole messiness of the analysis played out with me as a participant, which is not the way these things are usually done. But it worked out okay in the end.
And to jump a bit, so from the point of view of technology and technological development, recently we've spoken to quite a few people who do imaging heavy work for EMBO and for Review Commons and for previous episodes of the podcast. And there's when we ask them what is it that they can't do right now that they would like to do. So what are the limits technology is placing on their work? A lot of them say that in practice, they're so tied up right now with the problems of properly analyzing the volumes of data that they're generating, and not just genomic data, but imaging data, movies, live imaging, that they are not yet focused on the forefront of where, for example, imaging has to be. And I know that one of the initiatives at CZI is a new imaging center. And so maybe you have a bit broader view here. Do you see, not at the analytical side, but are there things that you would like to do or hope to develop for biological imaging that are currently not possible?
Yes. Well, with our new imaging institute, we hope to exacerbate the problem you just mentioned. We have an initial grand challenge in cryo-electron tomography and have a sort of goal of trying to increase throughput in that field by 100-fold, as well as increasing resolution, all of which is going to make it possible to have even bigger, more complex data sets that are going to strain our attempts to understand them. And, we recognize that there will be other big problems around computation, and we're going to seek to address them, both within electron tomography, cryo-electron tomography, but also more broadly. And for example, our tech team at CZI has spent a lot of effort trying to support Napari, which is a community-built interactive multidimensional image viewer for Python. And it's designed for browsing, annotating, and analyzing large multidimensional images. Actually, one of the founders of that came from CZ BioHub, Loic Royer, who is a terrific young scientist. And CZI has taken on, trying to help grow that ecosystem. And so it's a big effort on our tech team to help make that available for folks to try to ease the bottleneck you're talking about, which is a real one.
But are there things at a really fundamental level that you would just like to see that you can't see right now at the data generation and not the data crunch and the pipeline for analyzing large volumes?
Well, yeah, I mean, the things you'd like to see, and this is the point of going to cryo-electron tomography, we'd like to see and identify all the proteins in a given cell or a large fraction of them and identify them. I mean, right now you look at the tomograms and you can identify the really big things like the ribosomes and whatnot. But our folks have calculated that, you should be able to identify about half the proteins, the heavier, larger molecular weight half. And that's like within sight within the next decade. So, we're going to want to try to see those things inside cells.
CZI's mission includes a commitment to open science. They support the development of infrastructure and platforms to make open data sharing possible. CZI supports projects aimed at, quote, shifting incentives towards rewarding open science practices in biomedical research, unquote. Shifting incentives towards open science is something EMBO is also committed to, most recently by making refereed preprints equivalent to peer-reviewed journal articles in the evaluation of postdoc fellowship applications. I asked Dr. Quake what role funders can play and what kind of incentives they can establish to promote open science.
Well, I mean, you can require use of preprints, right? And that's sort of what we did at the BioHub when we started. We said all our research and everyone we fund from the BioHub is going to be required to post their work on a preprint server on the same day they submit it to a peer-reviewed journal. And I think we were the first organization to actually require that as a matter of policy. And that was like 2016. And so it was, not clear that people were going to be willing to do that. And we were all kind of holding our breaths a little bit, saying, this is kind of a big ask of people. And they're like unsure about it. They're worried about getting scooped and all kinds of other things. And at the end of the day, it was very successful. We had pretty good compliance, large fraction of people willing to do it. Nobody turned down our money because they didn't want to do it. And, we never found an instance of somebody not getting a job because, they had posted a preprint and gotten scooped on it. And, we'd made a bunch of promises that we'd help those folks out if it ever happened. And we were never called upon to do that.
Okay, but you had you had the prospect of a safety net if it was required.
Exactly. We did. Then the tidal wave hit, which was the pandemic. And then we were just ahead of that whole wave coming. And I think the pandemic really transformed how people use preprints, and it became normalized in biology in a way that was amazing and kind of accelerated. And we're continuing to see that happen. CZI has similar policies about requiring its grantees to use preprints. And that's been, I think, an important role for funders, to answer your question where you started with. You can say, this is the norm, and we expect you to do this. And if you want to receive our grants, you have to play in that.
So we're arriving somewhere where, and also now it will be mandated in the US in the near future, at least for papers within the US system, that there will have to be a preprint or an open version. But of course, that's not the same as the open data. The open science component of it is different from open access, right? So that requires a lot more storage capacity, among other things. Is that something that CZI is actively involved in?
We're thinking about that going forward. I wouldn't say we have anything that I can really share at this time to be exciting, but I will point out, as far as sharing data goes, that genomics has really led the way for that. And, you've got to give credit to the journals, because they all began requiring that anyone who published a paper in genomics, all the sequence data, had to upload their raw data to, a shared server. And that wasn't a thing before in biology. And I think, that was a really transformative moment, and sort of set of policies that, hey, and it's great from my view. And look at my students and postdocs, you have much less incentive to overinterpret your data if you know that someone else is going to download your raw data and reanalyze it. And it creates a certain virtuous circle of intellectual honesty in the field that I think has been very positive. So journals requiring people to upload their data has been great. It started in genomics. I think you're going to see it in microscopy and other areas now, and that's going to percolate through the field in a way that's going to be terrific. And just to come back to the thing you raised, the question is, when do you share the data a little bit?
Right.
And what we did at the BioHub for our big projects, we decided we were going to share all the data when the preprint went up. And so we made a point of taking a risk on our side that other people would analyze our data and scoop us with it. And we put all the raw data up for our tabular projects as soon as the preprint went up. And we asked people, please, treat this with respect and, use it, and it's going to help you do something else, go ahead. And this was driven a little bit by, our decision in this field, in this area, by Mark and Priscilla's desire that their philanthropy be used to accelerate science. And how do you accelerate science? And I think a big part of it is sharing your results as quickly as possible and eliminating publication delays. That's the value of preprints, because it takes a year and a half to publish something. That's a delay in other people learning about your discovery. And so we took the same philosophy to data. We'd share the data as soon as the preprint went up. And there were beautiful examples, especially with the first Tabula paper, of people who used that data and were able to publish other things that were super interesting and important because we had shared our data ahead of our publication. Their papers came out ahead of ours. So, for example, on two of them very specifically, RNA velocity, and Sten Linnerson and Peter Karchenko, they used our data to show that the RNA velocity algorithm worked across a wide variety of cell types. And they did that because a referee asked them to do that. And so they needed to do it to get the paper into Nature. And Sten was very clear about that it was important to them that they had access to that. And we thought that was great. Another one was Jay Shinduri, who was publishing an atlas on mouse of chromatin atlas. And he wanted to show, again, probably because a referee asked, that the chromatin and the RNA data would line up. So he used our RNA data to validate the chromatin results. And again, beautiful paper. I think that one might have been in Cell. But again, both of those came out before our Tabula paper did. They used our data. And great examples of how sharing data can help accelerate science.
And you mentioned a couple of times in your answer just now the value that was added by the peer review of the preprint, right? Because this is also a big discussion now, is there's a whole spectrum of the radical, just publish and have it commented, all the way to more conservative. Let's stick with the traditional paper when it's ready and has a stamp of a journal. In this case, how do you feel about the open peer review process? So making the peer reviews and the author responses and so forth available together with the manuscript.
Yeah, I've wanted to do that for years. I get these rejections and these reviews that really piss me off. It's like, how can a journal tolerate this quality of review? I should just post this review on the internet, because you see how superficial it is, and other people can judge. So, anyone who's gotten a bad review, I think, is philosophically very aligned with that view. I don't know how practical it is at the end of the day. And, I would say, just more generally, I think the value of preprints is to experts in the field, because you have your own basis to judge the quality of a result or a paper, and whether the authors made mistakes or pitfalls or something they didn't see. And the further away you get from having expert domain knowledge about an area, the less useful the preprints are, because you're not in a position to evaluate them. And the value of peer review is helping you, when you're a non-expert, have confidence in a result. And to the extent that publishing the reviews helps that, maybe that's a good thing. I don't think it's a full substitute. And like this idea that there doesn't need to be any quality control, I struggle with that a lot. So I see an enormous amount of value
Yeah.
in both preprints and full peer-reviewed paper. And where you sit in the middle ground, I'm a little less sure.
I think with and I believe your lab has used review commons, for example, for some recent work on B cell development, and I think there the part of the idea was that in between and what happened in practice is in between the preprint and the paper, you have first a revised manuscript, which often changes a lot. So one of the things we've been looking at is the change between the original submission, the revised preprint, and then the published paper. And so far, it looks like not a lot changes in terms of data between the revised manuscript and the final published paper. It changes quite a bit between the peer-reviewed and the non-peer-reviewed version. But what happened there really was that about 60% of the time it took for the average paper to become a paper, which is when you would the average reader would be able to see the revised content, evaporated, so that it was down to like 70 days or something for the refereed preprint to be posted. So that if you were really in a bind, if you were trying to get into a new field, or if you didn't have a new model organism, and this kind of situation you mentioned of, well, if it's your field, you read the preprint and you'll know how good it is and so forth. But particularly, for example, with the pandemic, it happened that a lot of people became interested in a lot of things that they didn't necessarily have great expertise on. And that put a magnifying glass, I think, on the value of having some kind of quickly publicly available and curated peer review process. I think people underestimate how important it is to find the right reviewers, to police conflicts of interest, to make sure that the person commenting on the statistics is not the anatomist, and the person commenting on the anatomy is not the statistician, or that they can, but at least you know where the comments are coming from in each case. And this it's a little bit of a hybrid creature, and but it has, I think, been valuable in some ways as an experiment. The question is, can it scale up? Is I think one of the problems right now. But in your case, since you used it, or it was used in the lab, do you think the refereed preprint was an improvement over the original manuscript?
Well, it's funny you bring it up, because I'm meeting with that student later today to talk about our next step on that manuscript. So a little bit, the jury's still out for me. And, it's an experiment. This is like the first one we're doing here. And I'm curious as anyone to see how it's going to turn out. So I'm reserving judgment until we get to the end of the whole thing. I think for a lot of this stuff, I defer to the wishes of my students and postdocs about where they want to publish and what route they want to go down. And Michael, who's the student here, wanted to explore this, and I'm game. Let's try it, and we'll see where it goes. So we'll see. I'm glad we're trying it. Let's put it that way, because I'm a scientist. I like to do experiments. The landscape of publishing is evolving, and the way you figure this stuff out is by kind of jumping in. And Michael is the first one in my lab to be brave enough to try it. So kudos to him.
If you want to read the recent refereed preprint submitted to Review Commons by Steve Quake, first author Michael Swift, and Felix Horns, it's called Fate Bias and Transcriptional Memory of Human B Cells. You can find it at Review Commons or bioRxiv. It's a refereed preprint, so you can read not only the manuscript, but also the full set of referee comments and the author's response.
You wrote a few years ago in the New York Times, even though you're an optimist, you managed to, I think, find a more grim expression than publish or perish, which was funding or famine, which somehow makes it seem darker to me. And the idea that something has to change in this constant pressure research is under to pay, as you mentioned in the Times piece, not just the research funds, but their own salary and everyone who works for them and so on. And as many people have commented, this has often a pretty stifling effect on innovation and risk-taking and so forth. And so at CZI as a funder, how are you taking on this aspect of science funding? Well,
first, kudos to you for digging way back in history and finding those old New York Times things. And I'll give a quick shout out to my old friend Olivia Judson, who was the regular science columnist, and she went on sabbatical and got people to fill in for like a month at a time. And I was one of the months filling in. I only had to write 4 things. And, I got a lot of respect for journalists after that. and people who write regular columns. The first one was really easy to write. Second one was pretty easy. Third and fourth, I was really working to come up with material. I'm like, wow, how do people do this every week on a deadline? And that was a really interesting lesson to me. And yeah, in one of the columns, I did use the opportunity to say people are on this hamster wheel of funding, and especially in the U.S., where there's sort of a lack of reliable long-term funding. People spend an inordinate amount of time worrying about funding for their labs and not doing science. And they tend to over-apply for grants because if you ever lose one, you got to have another one to back it up. And so it creates this extra inefficiency in both the system and in people's professional lives. And I was kind of speculating, wouldn't it be nice if most people could just count on a baseline level of support? And I think most academics would be happy to run a group, which is like 2 students, right? And just work on what they wanted and be able to focus on science and be at the bench and not be spending all big portion of their time writing grants. And there's some fraction of very ambitious people that want bigger groups, and they'll jump on the grant thing and do that. But it felt like there'd be an overall improvement in efficiency if most people just had long-term support at a modest level. That was my proposal then. And I'm standing by that. I still think that's a good idea. It's not the role of CZI to do that, simply because even though we are a very large philanthropy, we pale in size compared to public funding of research. I mean, of the NIH budget, we're like 1% or something like that. And so we just don't have the scale to do that. We live in a world where the vast majority of funding of science is going to come from the government, and philanthropy does different things, right? I mean, we're never going to replace that. And it's sort of this question of how you allocate that funding is one that's going to be decided by the government and the agencies. And I hope they go back and take a look at that column too, because I think it's a model worth exploring.
And one last question on CZI and its mission. So we've talked a lot about what it's doing in the U.S. and both as BioHub and as a funder and as an innovator or innovative institution. Are there any international projects from CZI? Since we are a European organization, are they all for the United States academic and research community, or do you have any international programs?
We fund science globally. We have grantees from some large number of countries. I don't remember what the number is right now, but it's substantial. We're supporting all kinds of things in Europe, including projects. We fund individual investigators all over the continent, but some of the bigger projects we're supporting a big imaging study at the UK Biobank. We're supporting a big X-ray tomography project at the Synchrotron ESRC in Grenoble. Yep. Those are 2 big ones that come to mind right away that are pretty substantial investments on our part, as well as just numerous individual investigators across the areas we fund. I mean, you look at what we fund in the single cell genomics area, the cell atlases, and it's people like Sarah Teichmann at Cambridge and at the EBI.
Speaking to Olivia Judson and other scientists who have published successful books for a broad readership, one thing they invariably point out is that you need an agent. The odds that a feisty intern at Random House is going to pick your manuscript out of a pile and champion it are, to say the least, not great. Steve Quake is also an entrepreneur who has founded or co-founded several companies, including Fluidigm, Immumetrics, and Helicos. We asked him what advice he'd give researchers who are thinking of heading down a similar path.
I do get that question fairly frequently by students and postdocs who are considering a career as an entrepreneur in the biotech area. And I tell them something sort of similar, not to get an agent, but to find a mentor. And, biotech is not something you can do in your dorm room, right? I mean, it is a complicated, highly regulated, capital-intensive area. There's a ton to learn about how to do it right that we don't teach in universities. And so I encourage young folks who are interested in being entrepreneurs, take that first job at a venture-funded startup where you have professional investors, professional management who've done it before. Go to a young company that's just getting started, because then you see the whole landscape of what goes into it, and use that as like an apprenticeship to really learn how to do it before you strike out on your own.
The Chan Zuckerberg Initiative has funded grantees in 31 countries, including international teams in their ancestry networks for the Human Cell Atlas and expanding global access to bioimaging projects. For information on open calls, visit chanzuckerberg.com. To know more about the Quake Lab's research, visit their page at stanford.edu. To know more about Steve Quake, have a look at Clinical Assessment Incorporating a Personal Genome, published in The Lancet in 2010. For more on refereed preprints, to browse a collection, or to submit your own manuscript, go to reviewcommons.org. On EMBO's site, you'll find information about the postdoc fellowship application process, including how preprints and refereed preprints are evaluated. Thank you for listening to the EMBO podcast.